Antibiotics are usually secreted by competing bacteria in order to keep other bacteria from encroaching upon their territory so that bacteria have more room to prosper and divide. Now, bacteria want to be able to get around this dangerous uh, substance that the antibiotic is because it disrupts its normal function. Say it could destroy their cell wall entirely or something important for it being able to live. So it finds genetic material that is resistant to the antibiotic, and it tries to incorporate it into its own genetic structure so it can express resistance to the antibiotic. So, in this example, genetic material is present from previously lysed bacteria, and the bacteria is going to transform the DNA by incorporating it into its genome so that it can express resistance to this antibiotic. So, in this case, the resistant strain removes the ability for the antibiotic to bind to the receptor, and it gains antibiotic resistance. Antibiotic resistance usually disables the antibiotic from being effective upon the bacteria. Now, exponential growth on the MCAT deals with the process of binary fission in which a cell becomes two cells, and two cells become four cells, and four cells become eight cells. You'll usually either find questions being asked either in how many cells will exist after a certain length of time, or how many divisions did the bacteria undergo during a specific amount of time. So at period 0, you have what you start with. After one division, it's twice as much. After 2, it's twice as much as the previous, so on and so forth. So we come to the equation 2 to the n power, where n represents the number of divisions that bacteria underwent during this binary fission process. So let me give you an example that is very useful in microbiology. E. coli is considered the workhorse of microbiology. So if E. coli doubles roughly every 20 minutes, and it goes like this for an entire hour, how many times did the one E. coli cell divide? Well, one thing you can do when it comes to a lot of MCAT problems is use dimensional analysis, where you compare the minutes to hours and so on and so forth to arrive at your answer. In this case, 20 minutes goes into an hour 3 times. I should have flipped that. And you'll come out with 3 divisions. So the one E. coli cell becomes 8 E. coli cells after it is divided 3 times within that hour. Alright, moving on. So one bacteria becomes 2 to the n power for however many times it is divided. In this case, it is 2 to the 3rd power because it divided every 20 minutes. So at 0 time, you have whatever you start with, whether it's 1 bacteria or 1 million bacteria. And after the first division, which is 20 minutes in this case, it can be any length of time that the MCAT says it takes to divide. So it goes 1 division at 20 minutes, 2 divisions at 40 minutes, and 3 divisions by 60 minutes. So it'll be 8 times whatever you start. So multiply it by 2n to get your answer. Alright, anaerobic and aerobic bacteria. When you think of anaerobic, think of without oxygen. And when you think of aerobic, think with oxygen. If you have trouble remember it, think about how an A is usually placed before something to mean not. In this case, and, because they didn't want to do the double A's. So A equals not, or and in this case equals not, or not oxygen, or without oxygen. Here I'm drawing some test tubes where bacteria have grown in suspensions. Now the top is where there's exposure between the solution and the air, and at the bottom the oxygen can't quite reach it. So when the bacteria are growing at the top, they're aerobic obligate organisms. When they're growing only on the bottom, they're obligate anaerobic organisms. Obligate means that there's no mixture. It means they're not facultative. They don't do both. So bacteria can be either parasitic, symbiotic, or have no companionship. Parasitic is when the bacteria benefits and it harms you. That describes pretty much all pathogens. And when the bacteria is symbiotic, it benefits itself as well as yourself, and you can consider most of the body's flora that we need to survive symbiotic, because it prevents a whole number of problems, say from pathogens infecting us or enabling digestion of food, among other things. Now, bacteria move by a process known as chemotaxis. It kind of guides them in the direction they're wanting to go. And they tend to maneuver along concentration gradients for whatever they're wanting to get near. Here I'm drawing a solution where the more dots represents a high concentration of solute, and the bacteria will either move towards the high concentration or away depending upon whatever it's sensing, and it'll usually use a flagella or cilia or something along those lines to get to the concentration gradient that it desires to move towards. Now, moving on to the topic of transformation. We discussed this a little when we were talking about antibiotic resistance. Transformation, conjugation, transduction are pretty much the three things you're going to need to know as far as bacteria acquiring genetic information. In transformation, it acquires genetic material that has been left lying around and just incorporates it into its genome. Say it collects antibiotic resistant strains so it can protect itself from antibiotics. And in the case of conjugation, there's a pilus in which it bridges between the bacteria, and it transfers genetic information. The pilus itself just orients it towards it so it can share the genetic information between the genome. Now, in the case of transduction, viruses are involved. Say, for instance, a bacteriophage in its syringe injects its genetic material into the bacteria. Well, the material will incorporate itself into the genome, and the bacteria will express itself in future generations as it undergoes actually only in the case where it undergoes the lysogenic cycle, because it'll literally replicate itself continually with the virus material being expressed as it has incorporated into the genetic material. So every time it divides, it'll carry on the virus incorporated genetic material.